Hello everyone and welcome to my channel EG Life Science. Here we are going to discuss about the MCQ in a biochemistry, and these MCQ will be helpful in a set life science exam. So please subscribe my channel EG Life Science, like my video and share it to your friends. So let's start. The first question is the term biochemistry was first introduced by. Four options are there: Karl Erking, B option Karl Neuberg, J Sumner, and D option is a Swedberg. The correct answer for this question is a Karl Neuberg. This scientist, he was a German chemist. Also, the another scientist, that is A. E. Garrod, he was known as a father of biochemistry. The other options like Karl Erking, J. Sumner, and Swedberg. The Karl Erking, this scientist coined a term that is a biotechnology. So he is known as a father of biotechnology. J. Sumner, this scientist discovered that enzyme can be crystallized. Swedberg, option D, that is a Swedberg. The Swedberg unit is a non-SI metric unit for a sedimentation coefficient, and this unit is given by a scientist Swedberg, and this measures a particle size indirectly based on its sedimentation rate. One Swedberg unit is equal to 10 raised to minus 30 seconds. So the correct answer for question number 1 is a Karl Neuberg. Then moving to the next question, the first protein sequenced by Friedrich Sanger is, the correct answer for this is option B, that is insulin. The first protein sequenced by Friedrich Sanger was insulin, and the insulin, this insulin is a hormone. It is a protein hormone. It controls glucose level in blood. Next question, nucleic acid in a nuclei of first cell were firstly discovered by. Four options are there: Anthony Lavoisier, Mayer, Helmholtz, and Friedrich Mischer. The correct answer for this question is option D, that is Friedrich Mischer. The Friedrich Mischer observed the nucleic acid in a nuclei of first cell. He observed that the first cells are with a phosphorus rich chemical, and he gave a name it as a nuclein. The other option, that is Anthony Lavoisier, Mayer, and Helmholtz, has Anthony Lavoisier, he discovered that a role of oxygen in a combustion and a respiration, and he also proved the law of conservation. So correct answer for the question is Friedrich Mischer. Then the next question, the primary structure of a protein represents. Four options are there. The correct answer for this question is option A, that is linear sequence of amino acids joined by a peptide bond. We know that the proteins are made up of a number of amino acids, and these amino acids are joined to each other with the help of CONH bond or a peptide bond. So the primary structure resembles with the linear sequence of amino acids which are joined by a peptide bond, whereas the secondary structure refers to the regular local structure of the protein backbone. It is stabilized by intramolecular and sometimes intermolecular hydrogen bondings of amide groups. Two common types of secondary structure are there, that is alpha helix and beta plated sheath. The tertiary structure of a protein refers to the overall three-dimensional arrangement of its polypeptide chain in a pace. Generally, the tertiary structure is stabilized by outside polar hydrophilic hydrogen bonds and ionic bonds, and internal hydrophobic interaction between non-polar amino acid chain. So, the correct answer for this question is A. The primary structure of a protein represent a linear sequence of amino acids joined by a peptide bond. Then the next question, a tertiary structure of a protein is maintained by. The tertiary structure of a protein maintained by peptide bonds, hydrogen bonds and disulfide bonds. So the correct option for the question is option D, that is all of these. Tertiary structure maintained by peptide bonds, hydrogen bonds and disulfide bonds. Correct answer, that is option D. Then the next question, cobalt is a part of dash dash vitamin. Options are vitamin B1, vitamin B2, vitamin B3 and vitamin B12. The correct answer for this is option D. That is vitamin B12. Vitamin B12 is called as a cobalamin. It is water soluble vitamin and it acts as a cofactor in a DNA synthesis and in both fatty acids and amino acid metabolism. And because of a deficiency of vitamin B12, there is a pernicious anemia, and this is a autoimmune disease. Other option, that is vitamin B1, B2 and B3. The vitamin B1 is called as thiamine, vitamin B2 is called as riboflavin, vitamin B3 it is called as niacin, vitamin B5 that is a pantothenic acid, vitamin B6 that is a pyridoxine, vitamin B7 that is a biotin and vitamin B9 that is a folic acid or a folate. So the correct answer for the question is vitamin B12, where there is a presence of cobalt, or it is called as a cobalamin. Then the next question, process of fermentation is firstly recognized by. The answer of it is Louis Pasteur. The Louis Pasteur gave us a process of fermentation. The scientist Theodore Swan gave us a process of fermentation. Option B is the correct answer for this. The fermentation is a production of acids from a sugar. The process of fermentation is firstly recognized by a scientist Theodore Swan. Option B. The next question, vitamins were discovered by. The answer of it is option C, that is Casimir Funk. Casimir Funk in 1912 originally coined the term vitamine, and he is known as a father of vitamin therapy. The other option, that is Liebig. Liebig is well known for Liebig beef extract and a chemical fertilizer. So the correct answer for this question is option C, Casimir Funk. The next question, that showed that the viruses are nucleoprotein in nature. The answer of it is option C, that is Stanley. The Stanley or Wendell Stanley, he crystallized viruses and showed that viruses are a nucleoprotein in nature. One gram of fat gives dash dash energy. Answer for it is option D, that is 9 kilocalorie. Next question, fats are abundantly found in. The fats are abundantly found in option A, that is a reproductive tissue. The next question, which of the following lipid is abundantly found in a cell membrane? The answer of it is phospholipids. The phospholipids are present abundantly in a cell membrane. Cell membrane and internal membranes are made up of glycerophospholipids. The glycerophospholipid molecules are composed of glycerol, a phosphate group, and two fatty acids chain. So the answer of it is option C, that is a phospholipid. The next question, synthesis of glucose from fat is called as. Four options are there: glycolysis, Krebs cycle, saponification, and gluconeogenesis. The correct answer of it is gluconeogenesis. The gluconeogenesis is a pathway by which glucose is formed from non-hexose precursors such as fatty acids like glycerol, lactose, pyruvate, and glucogenic amino acids. These precursors or these non-hexose precursors are converted into a glucose. During a prolonged fast or a vigorous exercise, the glycogen stores become depleted and glucose must be synthesized in a de novo process to maintain a blood glucose levels. So the process of gluconeogenesis is essential for a synthesis of glucose from the fat or non-hexose precursors, and this process of gluconeogenesis occurs in a liver and kidney. And gluconeogenesis stimulated by a hormone, that is a glucagon, epinephrine, and cortisol. So the correct answer for the question is option D, that is a gluconeogenesis. Other options that are glycolysis, Krebs cycle, and saponification. The glycolysis, here there is a production of a pyruvic acid from a glucose and a generation of ATP in form of generation of ATP, and this is a anaerobic reaction. Krebs cycle, the Krebs cycle is occurs in a mitochondria. It is also called as a citric acid cycle, and there is a generation of FADH2, NADH2, ATP, and a hydrogen water molecule and a CO2. Then the option C, that is a saponification. Saponification means a formation of metabolic salt of fatty acids, and such a salt are called as a soap or a triglycerides. These triglycerides are combined with a strong base to form fatty acid metal salt during a soap making process. Means the option A, B, C are wrong options. The correct answer of this question is a gluconeogenesis. Hydrolysis of fats by alkalis into fatty acids and glycerol is called as. The answer of it is a saponification. Hydrolysis of fats by alkali into fatty acids and glycerol is called as a saponification, option B. The next question, beta oxidation takes place in. The beta oxidation is a catabolic process by which fatty acid molecules are broken down in a cytosol in case of prokaryotic organism, and in case of eukaryotic organism this beta oxidation occurs in a mitochondria, where during this beta oxidation fatty acid molecules are break down into a acyl coenzyme A, and this acyl coenzyme A, it enters into a TCA cycle. So beta oxidation takes place in case of eukaryotic organism, answer will be the mitochondria. In case of prokaryotic organism, answer will be the cytoplasm. So two answers are there. So this question is incomplete. In case of prokaryotic organism, answer should be B, that is cytoplasm. In case of eukaryotic organism, answer should be the A, that is mitochondria. So thank you very much. Please subscribe my channel and share it to your friends.